So we went through the cell life cycle, but I was going really fast. So let's just take a look at it again. And I mentioned last time that we go from G1. This is where the normal cell function, the cell is just working fine, started to grow, started to duplicate the organelles, the proteins, and other things, but not the DNA. This is for the G1. So the cell is functional, just everything is going normal, and it starts to double the organelles and protein synthesis. Nothing about the DNA yet. So the S phase, which is the next phase, this is the one that the DNA replicate. And the S is coming from synthesis. S, the first letter of synthesis. So it means DNA and histone synthesis. So G1 and S, both of them have something that's duplicating and being synthesized, but G1 is everything but the DNA, and the DNA is in the S phase, which take about 6 to 8 hours. And then go to G2, which is the protein synthesis that follow the DNA synthesis, and then you start the process of mitosis, which is the actual division. Okay? So we are at G1, normal, start to duplicate things, organelles and others. S, duplicating the DNA, DNA synthesis, S, and then G2, protein synthesis. And then you go to mitosis. Mitosis itself is prophase, metaphase, anaphase, and telophase. And that would be followed by cytokinesis. So mitosis and cytokinesis. And then after mitosis, after the cell divides and it gives the number that's needed at this time, the cell can go to G0, which is if the cell is specialized, it will spend some time doing the specialty, producing hormones or growing something and so on. It can stay there or it can move again. So normally it will move again to G1, S, G2, mitosis, cytokinesis, G1, S, G2, mitosis, cytokinesis, and the circle goes on and on. So we call everything but mitosis, it's called the interphase, which is between the phases. G0, G1, S, G2. This is all together is called interphase. So S phase is a DNA replication, and it is always important to remember the enzyme that catalyze anything, synthesis or any reaction. So it's always important to remember that. So which enzyme is the one that duplicate the DNA, make a copy of DNA, which is DNA to DNA, DNA polymerase. What's the one that makes RNA from DNA?
RNA polymerase?
Yes. So enzymes are always important. So interphase, S phase, S is coming from DNA synthesis by DNA polymerase. You start by unwinding. Of course, the codes are inside, buried inside. There is no way that you can see it and make a copy of it before you unwind, so that you expose the codes and you can make a copy out of it. Okay? This is like the instructions in a paper in an envelope. You have to open it first, you have to unfold the paper, and then you can copy it, right? So unfolding first, and then the DNA will go base by base and making a copy, and this is called DNA replication. Mitosis itself, I mentioned before that mitos. Where is mitosis coming from? It's coming from the Greek mitos. And mitos means thread, threading. So mitosis is threading. What threading means? These threads that we will see that will align and attach itself to the chromosomes that are doubled. And then when it contracts, it will separate the two sets of chromosomes, one to this side and the other one to this side. So this is where the name came from. Threads attach and pull away the two sets of DNA after being duplicated. So in mitosis, DNA will be You will make double the amount, which is 2 sets of chromosomes, and the DNA will be tightly coiled. What do you call it if it is loosely coiled? Yeah, chromosomes. Chromosomes loosely tied. Chromatin tightly tied. What's the difference? No difference. Both of them are DNA wrapped around histone. How is it wrapped around? Is it tight? Is it loose? That's it. That's the only difference. If it is loose, you call it chromosome. If it is tight, you call it chromatid. Chromatin. And in the middle, there is a centromere that's connected to the chromatids, and the protein complex around the centromere is called kinetochore. And you go from prophase, metaphase. What's the next? Anaphase. And what's after?
Telophase.
Okay. Let's understand what does it mean. Prophase. Prophase means the first phase, or the preparation phase, the first phase, the beginning. Pro. This is where the nucleolus will disappear, and centrioles, pair, microtubules, and the nuclear envelope will disappear. And this is important. Specifically, nuclei, the nucleus or the nucleus disappear, and the nuclear itself membrane disappear. So now the content of the nucleus is exposed, right? The nuclear envelope will disappear. So now you have the chromosomes loose. What are you going to do next? Metaphase. And meta, translation, it's Greek, that means equator, which means you will arrange the chromosomes along the equator. This is where everything makes sense in physiology. So always remember this. There should be a reason why they call it this name. So metaphase is when you arrange the chromosomes along the equator of the cell. And that's why it's called the metaphase or metaphase plate. So this is the interphase. If you look at this, right before that in the interphase, you see actually an envelope around the nucleus, right? Surrounding. At this point, you don't see a nucleus. I'm sorry, you don't see the nuclear envelope. It disappears. The surrounding of the nucleus. It will disappear. Metaphase, this is the equator. Do you see this? This is the arrangement of the chromosomes. This is what's coming from one side, and this is coming from the other side. So the red is one set, the black is one set, and did you notice something? Red and black is not all red or all black, right? It is red, black, red, black, red, black. Because you have two copies, you're not going to just separate. This is the original one, you make a copy of it. You will not separate the copy in one side and the original in one side. You don't do that. You mix them, and you take red, black here, okay? So let's say the black is the original one, and you make a copy that's red. You are not going to separate the black in one cell, the new cell, and the red in one cell. It doesn't go like this. You double it first, and then you mix it together, and you take half here mixed and half here mixed, okay? So you put it around the equator, and this is a microscopic picture showing that. Here is the equator, and this is the centriole. These are the threads, which are the microtubules attached to the chromosome of the DNA in the middle. And then anaphase. Ana means pulling away, or away. Okay, translation, I'm just translating right now. Ana means away, from Greek to English. So anaphase means the microtubule threads will after attach itself, it will contract, pulling away. And this is where the name came from, anaphase. Away, the phase that the chromosomes move away from each other. Telophase. What's telo means? Why did they call the telephone? Where the name came from? Did you ever think about this? Telo or telephone. Phone means sound. Translation. Tele means away. So telephone is sound away. The sound that's transferred away. You are here, somebody else in New York, and the sound transfer or the sound go from here to there, right? Telephone. Tele means far away. Okay? Or distance. It means distance. Phone means sound. Sound in a distance. And this is where the name came from. So tele means distance. This is where this set is in one side and the other set in the other side. And this is where the name came from. Telophase. And in telophase, after you move the two components away, one on this side and the other one on the other side, the membrane will reform again. And the nucleolus will reappear again. And you have two cells now. So if you look at this, this is the anaphase. And ana means away. They pull away. They are not distant yet. They just start to pull away. And away means ana. Anaphase. So anaphase, telophase. Okay, so this is the anaphase. And cytokinesis. What's cyto and what's kinesis? Kinetic. When what's kinetic? Movement? Movement? Yes. Cytokinesis means the cell movement. Okay, so this is where the cleavage furrow around the metaphase and the cells move away from each other, giving you the two daughter cells. So this is the telophase, and you see how they are distant from each other, and this is called the cleavage furrow, okay, which cleavage means separation or dividing, and furrow means depression like this, fissure, depression like this. So the cleavage furrow, and then it will separate into two parts that will start to move away, as you see. So this will be when you see two cells separate from each other, this is cytokinesis. The cells that have long life, of course, are cells like liver cells, neurons. They live for a long time. There are other cells like red blood cells will live for 120 days, 4 months. There are like epithelial cells will live for hours, maybe 1 day or something. Same for the gastric, the inside of the stomach. They will live for short time. So depending on the life, the cell life, if it is long or short, it will make the mitosis slow or fast. And this will need ATP. You have the ATP, you can divide. You don't have enough ATP, you slow down the division. The hardest of all is muscles and neurons. They rarely divide. So by default, if the muscles are gone or the neurons are gone, 99% of the time it's gone. There are some exceptions that we will talk about later on, but normally muscles are gone, it's gone. Okay? And you will say, okay, like if my muscles died, part of my muscle died, it's gone, and what's going to happen? You replace it by fibrous tissue, fibers, but no muscles. No, muscles die and muscles reappear. It doesn't go like this. Muscles die, that's it. You can't make other muscles. You replace them by something else that you always have, fibrous tissue. Same for the heart or any muscle. So what's the fast? The fastest of all, skin and digestive tract. It's always turnover, maybe more than one time per day, versus neurons, it stay lifelong. What promote or regulate the cell division? There is something called M-phase promoting factor, MPF, and other growth factors that come from outside, like hormones or something. These are the factors that promote or inhibit M-phase. That is, generally speaking, growth hormone will stimulate most of the cells. Prolactin will promote the division of which cells? Can somebody tell me? Prolactin, which cells are you promoting to grow?
Gland and duct cells.
Glands of what?
Memory.
Lectin, of the mammary glands. Yes, it promotes the mammary glands. Epidermal growth factor, what does it promote the growth of? From the name. Yeah, epidermis. Epidermal growth factor. The factor that promote the growth of epidermis, right? The name gave it away. Just think about it. Fibroblast growth factor, same thing. Erythropoietin for the red cells, thymosin for the thymus and the immune system, and so on. Now, if something happened abnormal, the cell dividing in an abnormal rate, like speeding up really fast, that will be tumors or cancer. And tumor, it can be benign or malignant. If it is malignant, we call it cancer. And the cancer go in steps. It's not like cells are completely normal, all of a sudden it's cancerous. It doesn't go like this. First of all, there is an abnormal growth. Okay, an abnormal growth means the DNA become crazy for some reason. There are a lot of reasons that can make that. You make some, you stimulated the DNA. Okay, you did something like you're exposed to radiation several times. And this will change the nature of the DNA. The DNA will become crazy and it start to divide really fast. And this will be followed by primary tumor. Primary tumor is the first tumor to appear. So DNA will make a tumor, local, something in spot, in cyto. And then later on, it can metastasize. And metastasize means move or send parts that detach itself from the primary tumor, go like to the blood or lymph or something and move around until it attach itself to somewhere else and start the same process. Like we hear about lung cancer. And then lung cancer, get a treatment for lung cancer. Let's scan around. It's too late. The lung cancer is growing enough, part of it detached itself, move to the blood or lymph, go around, and now it stuck itself in the bone. This is metastasis. And the bone, at this point, if it started in the lung, and the bone is secondary, so this is called the secondary tumor. So primary versus secondary versus metastasis, right? Primary metastasized to make secondary, basic. The other name for tumor is neoplasm. And what's neo? New. Neo means new. Very close. From Greek to English. Again, it's benign tumor and malignant tumor. What's the most important thing to determine is it benign or malignant? There are a lot of factors, but what's the most important? Benign tumor, it's not the size, by the way, just for your information. Maybe this is benign tumor and this is cancer. It's not the size. Okay? Is it contained, meaning is it surrounded, well defined? Okay, the cell is separated by itself, or it is within the tissue, not separated. So is it contained or invading? All right, like this. This is a tumor, large, and this is a tumor that's small. It doesn't mean this is cancer and this is benign. It has nothing to do with this. It goes like this. Is it contained like this, so you can take it out, or does it go like this? Okay. Does it invade around and irregular, or is it contained? The other thing is, can it send secondaries, like part of this detach and move in the blood, go somewhere else, let's say the bone, and go to make another? So does it metastasize? Never metastasize. The benign will never metastasize. Contained, invasive, metastasize, does not metastasize. There are, of course, other things, but this is the most important. Just to differentiate between those two. So this is how it goes. The number one, abnormal cell appear, start to divide abnormally, start to make other cells invade the surrounding, go to the blood or lymph, both of them are okay, until it goes to somewhere else, metastasize, going somewhere else, metastasize. And then it will make secondary tumors or secondary cancers. Cell differentiation. When the cells are differentiated, means the cells are given the time to differentiate. Differentiate meaning grow, become mature, become good working cell. This is normal. For malignancy, the cells are growing fast enough that you didn't give it the time to differentiate. So the cells will be undifferentiated. What's differentiation means? It means it's growing and become mature and having its own function, specific function. You gave it enough time. In the cancer, it's going fast enough that you don't give it the time to differentiate, so it will become undifferentiated. So this is the third important difference between the benign and malignant tumor. Differentiated, benign, undifferentiated is malignant. Okay? Why? Benign is slowly growing, so you still have the time for the cells to differentiate and grow and mature. While the malignant are going fast enough, you didn't give it the time to grow, mature, become specialized, differentiate. So differentiated, benign, undifferentiated will be the malignant. Okay, so this is for this chapter, and we will move to the next chapter.